Assalamu alaikum. The lecture is about the movements of different substances across the plasma membrane. You must be knowing that a proper exchange of solutes, solvents, ions is very vital for the normal functions of the cell. We, the human beings, we are being blessed with 300 trillion cells, and every cell survives because of its communication, exchange of different substances or molecules across the plasma membrane. So today's topic will be consisting of two lectures. The essential role, of the biomembrane is to allow the movements of all compounds necessary for normal function of the cell across the membrane barrier. These compounds include sugar, amino acids, fatty acids, steroids, cations, anions, and definitely water. These compounds must enter and leave the cell in orderly, that is very important, to maintain the normal functions of the cell. Movements of ions and water. A movement of ions is through the ion channels, and specific channels are for a particular ions. All ion channels are made up of transmembrane protein subunits which come close to each other to form a pore through which ions pass selectively. If you remember, we discussed the components of the plasma membrane. So plasma membrane is traversed by the transmembrane proteins, which is crossing the whole width of the plasma membrane. And that transmembrane proteins, their outermost portion and their innermost portion, those portions are hydrophilic, and the central one is hydrophobic, so it remains in the center. So when these two transmembrane proteins, they come close to each other, definitely they will form a channel, a pore, and that pore is necessary for the transportation of these ions. All channels have two types of gate, which are controlled by opening and closing. Definitely, for the movement of these ions, there must be a switch. And these types of the gates, these are two types: ligand gated and number two, voltage gated. Ligand gated, specific molecule attaches to the receptor and open the channel. Example is acetylcholine. Receptor is present in postsynaptic membrane. So what happens? Acetylcholine released from the presynaptic region binds with the binding site of postsynaptic region, which triggers opening of the channel and influx of the sodium. So the response we get is in the form of influx of the sodium when acetylcholine attaches with the receptor of postsynaptic region. Voltage-gated channel, whenever there is a change in the membrane potential, that gate might open to allow that particular electrolyte to move in or out. Water channels, they are called aquaporins. Aqua means water. Porins, pores or channels. Aquaporins. Aquaporins are specific for the movement of water. Movement of water by simple diffusion is enhanced by movement of water through water channels, which are composed of tetrameric transmembrane proteins called aquaporins. There are four types of different proteins with a slight change in the structure. For example, in RBCs and cells of collecting tubules of the kidney. So wherever there is excessive use of aquaporins or water channels are required, definitely the different types of aquaporins will be there. There are at least 13 different types of aquaporins up till now which have been identified. We abbreviate those capital AP1 to capital AP13. So 13 different types. So most of these aquaporins, different types, around 6 types, they are present in the kidney. Mutation in the gene encoding AP2 have been shown to be the cause of one of the nephrogenic diabetes insipidus. You know what the diabetes insipidus is? It is due to either there is no proper release of antidiuretic hormone, and antidiuretic hormone is released from the posterior pituitary. So either the release is less or the response is less. Response of the aquaporins, the site of action of ADH. So if the kidney, the tubule, the aquaporins AP2 which are present in the kidney, they are not responding to ADH properly. Although ADH is being released from the posterior pituitary at a normal pace, what will happen? There will be, as the name is showing, antidiuretic hormone. So what it does? It don't let the water to be wasted or to be excreted in the urine. So if antidiuretic hormone is not functioning, it will lead to polyuria. And because the cause of this diabetes insipidus is in the kidney, so we call it nephrogenic diabetes insipidus. And when the cause of this diabetes insipidus is due to less release of antidiuretic hormone from the posterior pituitary, that will be simply diabetes insipidus. So nephrogenic diabetes is an example of the mutation in the aquaporin in the kidney. Another example is congenital cataract may be due to mutation in one type of aquaporin. Among the 13 known types of aquaporins in mammals, 6 of these are located in the kidney. Now, the types of transportations for substances across the plasma membrane. We divide it into 3 categories: passive transport, active transport, and when we want to transport something in bulk flow. Passive transport comprises simple diffusion, facilitated diffusion, osmosis. While active transport, as the name is showing, it takes place at the expense of inertia. ATP is used for the transportation of the different ions or different molecules. Bulk flow, the 2 different types, the 2 ways to export or import something in bulk. When something is going to be moved into the cell, it is called endocytosis. It is endocytosis, from out to into the cell. And when something has to be exported from the cell out, that is exocytosis. You can remember exit, it starts with the EX, so exocytosis, something which has to be exported from the cell. So the movement is from in to out. So individually, if we go into some details of these different ways of transportation. Passive transport. In passive transport, substances cross the membrane by diffusion. Diffusion, what it is? Net movement of substances from an area of high concentration to low concentration. No energy is required. It is also called downhill transport. Now, the factors affecting the rate of diffusion, important to know. Diffusion gradient. More the diffusion gradient, more will be the rate of diffusion. You see, the steeper, the higher the rate. Size of the molecule, smaller the size, the higher the rate, and vice versa. Temperature, the higher the temperature, the higher the rate. Diffusion media, that is important. High rate of diffusion in gas as compared to the liquid, and high rate of diffusion in the liquid as compared to solid. Surface area is another very important factor. The larger the surface area, the higher will be the rate. Now, simple diffusion. Non-polar, hydrophobic, small molecules diffuse directly through the lipid bilayer. This is an important point to remember. Non-polar, water insoluble, hydrophobic, water hitting, small molecules diffuse directly through the lipid bilayer, because the lipid bilayer, the center of the lipid bilayer, does not allow polar, water soluble, hydrophilic, larger molecules, or even electrolytes, even the ions, they cannot pass through the plasma membrane. A reason I tell you later. A polar, hydrophobic, large substances cannot pass directly through the lipid bilayer, which is done by facilitated diffusion. Examples are water, ions, and carbohydrates. You can make your idea or make your concept regarding why does not plasma membrane allow all the substance to move into the cell. The center of the plasma membrane, this is the plasma membrane, which is a lipid bilayer. At the two extremes, inside and outside, these are the hydrophilic portion. The center of the lipid bilayer, that is hydrophobic. So only hydrophobic molecules can pass through this plasma membrane, this barrier, like small non-polar hydrophobic molecules like steroids, CO2, O2 can pass directly into the cell, while this membrane will not allow polar, which means water soluble, hydrophilic molecules, for example glucose, water, and ions. They need specific channels to move into the cell so that this barrier is bypassed. Facilitated diffusion. Facilitated diffusion is the movement of specific molecule or ions across the plasma membrane. Facilitated diffusion is assisted by either pore proteins or by carrier proteins, and the direction of the movement is down the concentration gradient of the molecule is concerned. Energy may or may not be required. I will tell you later with a slide how we differentiate between what is the difference between a simple diffusion and facilitated diffusion. So simply facilitated, as the name is showing, it is being facilitated by either pore proteins or carrier proteins. Pore proteins, charge ions such as sodium, potassium, calcium, and magnesium cannot diffuse across a non-polar center of the phospholipids bilayer. Pore proteins open up pores or channel across the membrane to allow entry or exit. Each pore or channel is specific and will allow only one particular type of ion through. This is the slide which is showing the actual difference between the simple diffusion and facilitated diffusion. In simple diffusion, it is just a movement because those smaller non-polar molecules can directly cross the plasma membrane, so there is no assistance of carrier proteins in simple diffusion, while in facilitated diffusion, carrier proteins are required. Speed is relatively slow. The speed here is fast because they are crossing the plasma membrane through a pore, a channel. It is not solute specific, simple diffusion, but facilitated diffusion is solute specific. Simple diffusion is always passive process, with no need of ATP as energy. While facilitated diffusion, in facilitated diffusion, energy may or may not be required. The process may be active or passive. In simple diffusion, allows passage of only small non-polar molecules, while this permits passage of large polar molecules. Another difference between the simple diffusion and facilitated diffusion, that simple diffusion is not affected by inhibitors, while in medical science, in medical field, we use inhibitors which affect the carrier proteins. So we can say facilitated diffusion is being targeted or affected by different inhibitors in medical science. So these are a few differences between the simple diffusion and facilitated diffusion. Facilitated diffusion may be passive as the simple diffusion is, or may be active. Carrier proteins, they allow larger polar molecules such as sugar and amino acids to pass through. A particular protein or polar molecule attaches itself to the binding site of a carrier protein. Then the carrier protein changes shape, and definitely for changing the shape, energy is required. Then the carrier protein changes shape and delivers the molecule across the plasma membrane. This is how the carrier proteins, they function. Osmosis, this is another type of passive transport. Osmosis is related with the diffusion of water only. Diffusion of water across a selectively permeable membrane. When we say selectively permeable membrane, that means that membrane allows only water to move in and out of the membrane. So all of you is very much familiar with the process of osmosis. So what should you expect? In osmosis, water moves from an area of high water concentration to an area of low water concentration. Is energy required? No, not at all. No energy is required. Water travels in and out of the cell through aquaporins. Now, the different terms are related with the osmosis, which are being used in different texts. Hypertonic, hypotonic, and isotonic solutions. These are the relative terms. If you place three chambers, the first one is having the salt concentration or sodium chloride concentration 0.9%, which is in fact is isotonic with the RBCs. 0.9%. In the second compartment, second chamber, you place a solution of 1.9% sodium chloride solution, and in third you place 2.9% of the sodium chloride solution. So the third chamber, if you compare with the second one, the third chamber, which is having 2.9% of sodium chloride, that is hypertonic as compared to the second chamber, which is containing 1.9%. And this second chamber, which is having 1.9% of sodium chloride, it is hypertonic as compared to the first one, which is having 0.9%. And if you move from other direction, 0.9% sodium chloride solution is hypotonic as compared to the solution which is present in the second chamber having 1.9% of sodium chloride, and the second chamber is hypotonic as compared to the third chamber, which is having the more sodium chloride concentration. So these are all the relative terms, whether isotonic, hypotonic and hypertonic solutions. This is an example how the osmosis takes place. The two wings of this tube, it is being separated in the center by a selectively permeable membrane. Selectively permeable membrane which allows only water to move from this side, left side, to the right side. No movement of solute. So here you can see the concentration of solute is high. Water concentration is less. This is hypertonic. Water will flow from this column to the right column. Process of osmosis starts right by the movement of water. It crosses a selective permeable membrane to the right wing, and the process of osmosis continues till the concentration of the solute concentration in both the chambers, it becomes equal. You can see here, at this stage, at equilibrium, osmosis is completed. Now, this water in the left side has the potential, has the force to move, to cross the semi-permeable membrane and to enter into the right side. That is basically the osmotic pressure. Or in other words, you can define osmotic pressure, because the process of osmosis starts right after you place these two types of solutions here. Selectively permeable membrane उसको separate करती है, तो ये process start हो जाएगा. Or another way to define osmotic pressure is you place a mechanical pressure at this column so much that the process of osmosis stops. So that pressure which is required to stop osmosis is osmotic pressure. This is what happens in the blood, to the blood cells, when they are placed in the different types of the solution, hypertonic and hypotonic solution. These are the normal dumbbell-shaped cells, RBCs, circulating in the blood. This dumbbell shape is, all of you are familiar with, that it is to increase the surface area, to get to receive more oxygen. So this dumbbell shape, if you placed RBCs in water or in a hypotonic solution, what will happen? Water will enter into the cell, into the RBCs, and we get this picture. They swell up until the time comes when they burst. So this is the reaction we get after we place the RBCs in a hypotonic solution or even in a pure water. आप सब स्टूडेंट्स ने फिजियोलॉजी में ऑस्मोटिक फ्रेजिलिटी का प्रैक्टिकल करते हैं, तो उसमें यही मैकेनिज्म आपको फॉलो करता है. So if these RBCs, they are placed in a hypertonic solution. Hypertonic solution में क्या है? Water concentration कम है. So water concentration RBCs में ज़्यादा है as compared to that hypertonic solution. So water will leave the RBCs and the RBCs will be shriveled, will shrink, and you get this type of picture of RBCs. So this is how the RBCs, they react when they place in different solutions, hypotonic or hypertonic solutions. Osmosis, so now you tell me, when a cell is placed in hypotonic solution, what will happen? Just recall, water concentration is in the cell, either it is less or high. Water flows in or out of the cell. You take your time. Tell me. Okay, don't get confused. Water concentration cell में it will be low. Water flows in or out of the cell. Water will flow into the cell. That is why when RBCs are placed in hypotonic solution, they swell because water moves into the cell. And opposite to that, when a cell is placed in hypertonic solution, what will happen? Water concentration is, what you assume? It is high or less in the cell, or water flows in or out of the cell. Recall the pictures which I showed you previously. Water concentration is high in the cell and water flows, yes, out of the cell. That is why when RBCs are placed in hypertonic solution, the RBCs are shriveled, they shrink. This is how we get the response of the cells when they are placed in hypo or hypertonic solutions. This is the diagram which is showing the effect of hypotonic solution and hypertonic solution. This is isotonic solution. Isotonic solution for the normal, to keep the normal shape of the RBCs is 0.9% sodium chloride. So in isotonic solution, 0.9% solution, 0.9% sodium chloride solution, RBCs, they maintain their normal shape. In hypotonic solution, they swell up or even they are lysed. Or in hypertonic solution, they shriveled or shrink. This is the response of the cells. This is the effect of hypotonic and hypertonic solutions on the cell. What summary we get after looking over these slides? When a cell is placed in hypotonic solution, cell gains water through osmosis, animal cell lysed, and plant cells become turgid or form. When a cell is placed in a hypertonic solution, cell loses water through osmosis, animal cell shrivels, plant cells plasmolysed. Plant cells here, when placed in hypotonic solution, it does not, the wall does not break up, or because the wall is stronger as compared to the animal plasma membrane. जो एनिमल सेल है उसकी प्लाज्मा मेम्ब्रेन है और यहाँ पे टाइट सेल वॉल है जिसकी वजह से इट बिकम्स टर्जिड, यूजुअली वो रप्चर नहीं होती. Osmotic pressure. Let's discuss osmotic pressure in my another lecture. And after completing this topic, movement of different substances across the plasma membrane, I will give you assignments or some homework to do, and I will check it personally. In the end, stay blessed. Take all the precautionary measures regarding coronavirus. Educate your neighbors, educate your parents. अल्लाह आपका हामी नासिर हो.